Hi guys, and Assalamualaikum warahmatullahi wabarakatuh. My name is KHAIRUL AHMAD and today I'm going to talk about our project biology form 5 for 2021. I'm very thank you to our classroom teacher is Siti Zubaidah binti Nasir and our biology teacher Siti SYAHIDAH binti Jantan. Today I'm going to talk about chapter 1 organization of plant tissue and growth. Okay, let's proceed. I'm going to talk about subtopic 1.1 organization of plant. Plant tissue is divided to two groups. Is it meristematic tissues and permanent tissues? Meristematic tissues is divided to two groups, apical meristematic and lateral meristematic. Permanent tissues is divided to three groups. Is it epidermal tissues, ground tissues and vascular tissues? Ground tissues divided to three another group. Is it parenchyma, sclerocyma and collenchyma? Vascular tissue is divided to two groups. Is it xylem and phloem? Okay. For subtopic 1.1, we're going to focus to a permanent tissue. Permanent tissue are mature tissue we have experienced or experiencing depreciation. There are three types of permanent tissue which include epidermal tissue, ground tissue and vascular tissue, each carrying out a different function. First one, we got the epidermal tissue. Epidermal tissue layer the outermost surface of stem, root of young plant. Epidermal cell wall which are exposed to the air have a waterproof. Epidermal cell wall which are exposed to the air have a waterproof layer called cuticle. The cuticle reduces loss of water through evaporation. Protect the leaf from mechanical injuries and pathogen. They are modified epidermal cell according to their function. Like gut cell, control the opening of the stomach, and root hair cell, increase the surface area of the root for water and mineral salt absorption. Okay. Secondly, we got the ground tissues. First, we got a parenchyma tissues. Parenchyma tissue are simplest living cell and do not undergo differentiation. They have the thinnest cell walls. Parenchyma tissue are always in a turgid state, providing and maintaining the shape of herbaceous plant. Involved in photosynthesis, help in storage of starch and sugar, and involved in gaseous exchange. Involved in the repair and regeneration of plant tissue as well as in the vascular system. Secondly, we got a collenchyma tissues. Collenchyma tissue are made of living cells with mature cells that are flexible. Have cell walls made of pectin and hemicellulose. Their cell walls are thicker than the parenchyma tissues, provide mechanical support and elasticity to plants. The last one, we got the sclerocyma tissues. Consists of dead cells when they are mature. Their cell wall are the thickest among the three group tissues. Provide support and mechanical strength to the part of mature plants. These tissue also help in transport of water and nutrient in plant. Okay. Next one is vascular tissues that divide to two groups. Is it xylem and phloem? Xylem is made out of dead cell without the cytoplasm. The cell wall of xylem contain lignin. Consists of xylem vessels that are elongate, hollow and connected to each other from its root to the leaf. This enables xylem to transport water and mineral salt to all part of the plant. Okay, next one we got phloem. Phloem are made of companion cell and seed tube, made of living cell, which is the seed tube with the present cytoplasm. The seed tube do not have any organelles such as nucleus and ribosome as they decompose at mature stage. Phloem consists of seed tube arranged from end to the end, forming elongate and continued tube structure. Phloem transport sugar produced from the Phloem transport sugar produced from the photosynthesis from the leaf to storage organs such as root, fruits, and tube. Phloem transport sugar produced from the photosynthesis from the leaf to storage organs such as root, fruit, and tubers. Okay, that's all for subtopic 1.1. Thank you for watching.
Hello everyone, my name is RAESHAKTI SOKALINGAM, and today I'll be discussing about subtopic 1.2, which is meristematic tissues and growth. Okay, in these meristematic tissues, there are two components which are apical meristems and lateral meristems. In apical meristems, which is located at the tips of plant root and shoot, while for lateral meristem, which is consists of vascular cambium and cork cambium. In this diagram here, there are the shoot, stem, and root. Okay, for the zone of cell growth, there are three components which are zone of cell division, zone of cell elongation, and zone of cell differentiation. In the diagram below, there is the tip of the shoot, and the other diagram is the tip of the root. For this, it's from starting from the top to bottom. For the roots, it's starting from the down to top. The first is zone of cell differentiation. The second is zone of cell elongation. The third is zone of cell differentiation. For the root is just same, while placing is just different. The first, which is at the bottom, is meristematic zone. The second is elongation zone. The third is differentiation zone. Okay, now what is the characteristic of this tree? First is the zone of cell division. The first point is take place in apical meristem tissues, which consists actively dividing cells through mitosis. The second point, increase of numbers of cells cause the elongation of plant stem. 3. When new cells are forming, the other cells is pushed to the zone of cell elongation. Okay, for the second is zone of cell elongation. The first is consists of cells that are increasing in size. 2. Increase in size through mitosis and absorption of nutrients into cells and stored in the vacuoles. 3. A process called vacuolization. Vacuolization is where the small vacuoles will fuse into to form a large vacuole. 4 is diffuse water applying pressure against the cell walls, which pushes, elongates, and widens the cell. 3. Zone of cell differentiation consists of differentiation cells that differentiate once they have reached their maximum size. 2. Differentiate cells will form permanent cells such as epidermis, cortex, xylem, and phloem. The cells will change their shape and structures to be specialized cells in specific functions. For example, apical cells in leaves differentiate and form guard cells that controls the opening of stomata. On the other hand, epidermal cells in root differentiate and form root hair cells. So now is the type of growth. The first is primary growth. The primary growth that occurs after germination, and it takes place in all plants to elongate their stems and shoots. The first point takes place in the apical stems at the shoot tips and root tips. Second is starts when the meristematic cells in the zone of cell division of apical meristems are actively dividing, and is followed by elongation and differentiation of the plants. Third, at the shoot tip, leaf primordia and shoot primordia will grow to form new leaves and shoots, and enables the plant to increase in height. Whereas for the root tips, the root cap will become exhausted when they penetrate through the soil, and cause the cells of the root cap to be replaced by the meristem cells. In this picture below, which is quite blurry, apical meristem, which is primary growth, and lateral meristem, which is the secondary growth. So second is secondary growth, occurs mainly in eudicots and small numbers of monocots, shrubs, to increase the circumference of diameter of plant stem and roots. First point, will not have any woody plants such as herbaceous plants because they have soft tissues and generally have only one growing season. The result is from the division of lateral meristem cells, which is located at the stem and the root. Lateral meristems consist of vascular cambium and core cambium. So, vascular cambium, located between phloem and xylem tissues in vascular bundles. Core cambium, located under the epidermal layer. Okay, for the secondary growth at the stem. First, secondary growth starts when the vascular cambium divides actively through mitosis. In the cambium rings, divide inwards to form new xylem and outwards to form new phloem. The new xylem tissues become secondary xylem and the new phloem becomes secondary phloem. When undergoing secondary growth, primary xylem is pushed towards the pit, whereas primary phloem is pushed towards the epidermis. As a result, the primary xylem tissues are compressed to form a stronger wood layer. This is because the walls of xylems are thickened with lignin. The thickening of lignin provides mechanical support to plants. When the secondary xylem layer is compressed, there is an addition to the circumference of the stem, which causes the epidermis of the stem to stretch and crack. Cork cambium divides actively to form cork cells on the outer side and cortex on the inner side. The cortex layer protects the stem from insects and pathogens attack when the epidermis is cracked. Okay. Now for the secondary growth at the roots. Roots of plants also undergoes secondary growth in order to increase the circumference of roots. The process at the root is the same as the secondary growth of a endicott stem. Vascular cambium cells divide actively and combine to form a complete ring. The cells in cambium ring divide inward to form secondary xylem and outwards to form secondary phloem. Due to the vascular cambium activity, the roots becomes thicker. The cork cambium located under the epidermis divides actively to form cork cells. The cork cells provide protection to the root tissues. Which is in this picture given, there is A, B, C, and D, which is the 4 states of the secondary growth at the roots. Okay, now the necessity of primary growth and secondary growth. The necessity of primary growth allows maximum allocation of plant to absorb sunlight for photosynthesis. Primary phloem can transport the products of photosynthesis from the leaves to other parts of the plant. Whereas for the primary xylem can transport water and minerals from the soil via the roots to the leaves and provides support to herbaceous or young plants. The necessity of secondary growth provides stability to plants by increasing the stem and root diameters to suit the height of plants. provides mechanical support to plants, produce more xylem and phloem tissues, produces xylem and phloem tissues continuously to replace old and damaged xylem and phloem tissues, produces stronger and thicker bar to provide protection to the plant from excessive water loss, physical injuries, and pathogens infections, able to live longer by increasing the chances of seed production and reproduction. Able to live longer by increasing the chances of seed production and reproduction. Okay, the similarities between the secondary growth and primary growth. Both growths can increase the size of plants permanently. Both growths occurs in woody plants. Both growths involve cell division by osmosis. The differences. Primary growth. Apical meristem. Expect meristem tissues involved. Secondary growth. Lateral meristem. Vascular cambium and cork cambium. Primary growth occurs on stems and roots in younger regions of the plant. Aspect, part of the plant that undergoes growth. Secondary growth occurs when primary growth has ceased on matured stems and roots. Primary growth occurs longitudinally, direction of growth for aspects. Secondary growth occurs radially. Primary growth increase the length of stems and growth of plants. Aspect, growth effect. Secondary growth increase the thickness or circumference of stems and roots of plants. Primary growth, epidermis, cortex, and primary vascular tissues, primary xylem and primary phloem. Aspect, tissues and structures formed. Secondary growth, bark, periderm, cork cambium and cork tissues, lacticels, and secondary vascular tissues, secondary xylem and secondary phloem. Continuing, the presence of woody tissues. Do not have woody tissues for primary growth and have woody tissues for secondary growth. The thickness of bark, thin for primary growth, thick for secondary growth. Presence of annual growth rings. Absence of annual growth rings in primary growth. Presence of annual growth rings at the plant stems for the secondary growth. Now, the economic importance of plants that have undergone secondary growth. Plants that undergo secondary growth have strong and hard roots which are suitable to be used as structures for houseboats, furniture, fence, doors, and others. The presence of annual rings makes furniture looks attractive, and they can be made as furniture's decorative items. The wood and barks of some plants, such as Hopia sp, Merawan imbium, and Meranti also imbium, can produce resin, and oil. The substance can be commercialized and varnish, adhesive substance, perfume, and medicines. Flowering plants can be made as decorative plants as well. Commercialized fruits such as mangoes and mangosteen are the products of plants that have undergo secondary growth, and they are able to generate income as well as boost economic growth.
There are three types of plants based on life cycles, which is annual plants, biennial plants, and perennial plants. The first type of plant is annual plants. These plants only have one life cycle for a season or a year. They usually die after completing their biological cycle, which starts from germination and ends with flowering and producing seeds. Examples of annual plants are paddy plants, pea, and corn. The second type of plants is biennial plants. These plants take 2 years with 2 seasons of growth. The 2 seasons are vegetative growth and reproduction. Most of biennial plants grow at temperate regions. During winter, these plants will briefly stop the process, but during summer and spring, these plants will continue the growth process as preparation of reproduction by flowering. The plants will be able to flower and produce seeds and eventually dies. Examples of biennial plants are cabbage plant, carrot, and silver-fox cone. The last type of plant is perennial plant. These plants can live more than 2 years, depends on their species and conditions. These plants can be classified into 2 categories, which is woody perennial plants and herbaceous perennial plants. These plants can bear fruit and be able to flower many times throughout the years. This is because of the plant can change to their surroundings and the temperature change. Examples of perennial plants are potato plant and strawberries. Now, the growth curve in plants. The first growth curve are the growth curve of annual plants, which has sigmoid curve. This growth curve, it has 4 stages. The first stage is stage A, which is the decreasing dry mass. In this stage, the food is stored in the cotyledon. The food is used for germination before the leaves emerge to carry out photosynthesis. The stage B, which is the increasing of dry mass, is because the rate of growth increases rapidly. The rate of growth increases rapidly because the plant has carried out the photosynthesis. The third stage is stage C, where the dry mass is constant. The dry mass is constant because of the rate of growth is zero. In stage C, the plant is matured, and at the last stage is when the decreasing of dry mass. The decreasing of dry mass happens slowly because of aging, low rate of photosynthesis, shedding of leaves, flowers, and seed dispersal. The second growth curve is the growth curve of biennial plants, which has 2 sigmoid curves. The first growth season is when the plant produces leaves and photosynthesis takes place. After that, the food is stored in the tubers. In the second growth season, the food soil is used to produce flowers and seeds. The third and last growth curve is the growth curve of perennial plants, which has a series of sigmoid curves. Every year, the growth curve is a sigmoid curve. The rate of growth is high during spring and summer, but the rate of growth decreases during winter. That is all from us. Thank you for listening.